Before we jump into the bulk of this section, where we identify techniques in cultivating and identifying animal viruses, let's take a quick review of animations of replication of bacteriophages, as well as their various life cycles, as well as the lytic and lysogenic cycles.
The first step in the replication of virulent phage, such as T4, is attachment. Protein fibers at the end of the phage tail attach to specific receptors on the bacterial cell wall. In the penetration stage, the enzyme lysozyme, located in the tip of the phage tail, degrades a small portion of the cell wall. The DNA in the head of the phage is injected into the cell, and the phage coat remains on the outside. In the transcription stage, the phage DNA is transcribed into messenger RNA, which is then translated into phage proteins. Next, the phage DNA is replicated to make copies for new phage particles. The phage DNA is also the template for additional messenger RNA synthesis. During this stage, many copies of phage DNA are made, and the various phage proteins are synthesized. During the assembly or maturation phase, the phage DNA becomes packaged in the phage head protein, and the various components of the phage structure come together to form the intact or mature phage. Finally, a phage-induced lysozyme is synthesized. This enzyme digests the host cell wall, resulting in lysis of the host cell and release of the phage.
Now let's look at the lytic and lysogenic cycle.
When phage lambda infects E. coli, either the lytic or the lysogenic cycle may be followed. In both cases, the first step involves the phage attaching to the host cell and injecting its DNA into the host cell. In the lytic cycle, phage nucleic acid is replicated and phage genes are expressed, resulting in production of phage proteins. Mature phage particles assemble and the host cell lysis, releasing the phage particles. In the lysogenic cycle, the phage DNA is not replicated or transcribed. Instead, the phage DNA integrates into the host cell genome. The host cell can then replicate, carrying the integrated phage genome. The integrated DNA is referred to as prophage DNA, and the host cells carrying the prophage DNA are said to be in the lysogenic state. When the cells are exposed to ultraviolet light or to certain chemicals, phage induction occurs. The prophage DNA is excised, and the phage enters the lytic cycle.
Now let's take a look into how we might cultivate and identify some of the animal viruses. Initially, virologists were restricted to studying viruses in their natural hosts, which, as you could imagine, took a long time. Imagine the lifetime of a human. But since then, they've developed some in vivo, that is, in living system techniques, and some in vitro, as in a petri dish techniques, which help us cultivate and identify viruses more quickly. Why would we want to cultivate viruses? Well, first we need to isolate and identify the viruses in some of our clinical specimens. In addition, we might use viruses so that we could design vaccines to those viruses. Or we can do detailed research on viral structure, multiplication cycles, their genetics, and their effects on the host cells. We'll look at living animal inoculation, embryo inoculation, and cell or tissue culture techniques. Usually when we use live animal inoculation, we'll use specially bred strains of white mice, rats, hamsters, guinea pigs, and rabbits. Occasionally we'll use invertebrates or non-human primates. The animal will be exposed to the virus by injection in either the brain, blood, muscle, tissue, into a body cavity, into their skin, or on their foot pads. Here's an example of using a bird embryo. This is an in vivo situation. It's a living situation, as in the root vive. The egg is in nearly perfect condition because it's an enclosed situation that has all of the own nutrition that it needs. and so it makes a great environment for culturing viruses. We'll use chicken, duck, and turkey eggs most commonly. The egg is injected through the shell using sterile techniques. It can be injected in different parts of the egg depending on which virus is being studied and how we want to culture it. Another option is to use cell or tissue culture techniques. These are in vitro techniques. In vitro meaning in glass. Most viruses today are propagated in some sort of cell culture. The cultures must be developed and maintained. There are primary cultures and there are continuous cultures. Primary cultures are using freshly isolated animal tissue in the growth medium on a petri dish. These animal tissues undergo mitotic divisions to produce a monolayer across the growth medium. For these primary growth cultures, we can use embryonic, fetal, adult, and even cancerous tissues. Eventually, though, these cultures die off or mutate into a line of cells that can grow continuously. Animal cell cultures are grown in sterile chambers on special media. In a continuous culture, we'll see animal cells that have altered chromosome numbers that grow rapidly and show changes in morphology, and they can be continually subcultured to create new monolayers. By using continuous cultures, we can use cells that are genetically altered and can address a narrow host range. But then how do we view these cultures? We can view where viruses are in a culture by looking for clear zones. These zones of clearness contain lysed cells, or plaques, and they correspond to the area that contains viruses. We can use bacteria cultures, or monolayers, to identify bacteriophages, and then tissue cultures that we use to identify eukaryotic viruses. What are some of the medical importances of viruses? Well, infectious diseases are prevalent. Viruses are the most common cause of acute infections that do not involve hospitalization. So warts and herpes and influenza and chickenpox and shingles and herpes and such. Most of these do not result in death. However, some viral infections do result in high mortality rates. For example, AIDS, Ebola, or dengue fever. Also, we're starting to understand that there are some chronic afflictions that could be associated with viruses. For example, type 1 diabetes may have some association. Multiple sclerosis may also have a viral association, and certainly some cancers these days we're recognizing. Remember though, there are also beneficial properties in some viruses, so they're not all bad. In addition to viruses, we'll also see other non-cellular infectious agents. For example, spongiform encephalopathies or prion diseases. These seem to be caused by proteins that have no nucleic acid association. The mechanism of infection is not really very well understood. You may remember recently a big outbreak of mad cow disease in Europe. This is bovine spongiform encephalopathy. Occasionally we see similar symptoms in humans. For example, Creutzfeldt-Jakob disease. Another spongiform encephalopathy, where the tissues in the brain seem to resemble a sponge and contain protein fibrils. The signs of these diseases range from mental derangement to loss of muscle control and are progressive and universally fatal. We're not certain if they can be transferred from cows to humans or from deers to humans. This is one of those topics if you wanted to explore it in more detail for your final project. I'd be super excited. Satellite viruses are other infectious agents. These viruses seem to be dependent on other viruses for their replication. The adeno-associated virus was so named because it was thought to only replicate in cells that were affected with adenovirus. There are some plant pathogens, or viruses, that are really tiny. They're usually naked RNA, they're single-stranded circles, and they're really small. And somehow they seem to be able to fold the host's proteins. Again, some anomalies that you might explore further for final projects. Finally, let's look at how we treat some of these animal viral infections. Of course, antibiotics don't work because they're aimed at disrupting prokaryotes, so they don't work for viruses. There are several antiviral drugs, but most of them block virus function by targeting the function of the host cell, so you can imagine this causes many side effects. Most are also designed to target one of the steps in the viral life cycle that we learned about earlier in this chapter. Here are a few interesting antivirals. AZT is used to treat AIDS, and it targets the synthesis stage. There are integrase inhibitors. This is one that interrupts HIV's ability to incorporate its genetic information into the host's genome. Interferon is a product that we're starting to manufacture that's naturally produced by humans, and it can have some antiviral effects. And then again, there are vaccines that stimulate the immune system, like the influenza vaccine that you might have taken this year. So this brings us to the end of our unit. We've completed our survey of prokaryotes, eukaryotes, and viruses, and we've described some of the characteristics of different representatives. Now we'll move on to explore how microorganisms maintain themselves, beginning with their nutrition and then looking into their microbial metabolism.